Hello everyone, welcome to the next session of Oncology under 5 minutes by Nurture Oncology. Today we will be reviewing this article in brief. This article is on the topic tumor tissue detection using blood oxygen level dependent functional MRI based on independent component analysis. As we all know, glioma is one of the most common but lethal brain tumors. And accurate delineation of tumor tissue from the surrounding tissue maximizes resection without causing much deficit in a normal person. So, MRI is one of the commonly used pre-surgical imaging tool for localization and evaluation of the tumor entity. And the conventional options include T1, T2 and T2 fluid attenuated. There are limitations to this, as in diffuse lower grade gliomas have doubtedly invaded beyond the T1 contrast enhanced abnormalities and they might look a bit different. And then T2 imaging still struggles to differentiate the true tumor infiltrate from the edema area. Now, blood oxygen level dependent functional MRI is another tool for pre-surgical planning. Now, this tool basically characterizes activity-related change in the concentration of oxygenated and deoxygenated hemoglobin concentration, as well as fluctuation of cerebral blood fluid and volume. Previous bold fMRI studies have attempted to use resting state MRI to delineate brain tumors. Now let us get into what they actually did in this paper. Here they're talking about independent component analysis. What is this actually? This is basically a computational method used to separate multivariate signal into independent non-Gaussian components. So here, in the context of resting state functional MRI, ICA helps to identify distinct spatial patterns of brain activity by decomposing the functional MRI data into a set of independent components that represent different brain networks or sources of variability. I'll get into this in detail. That is, I'll simplify this. But before that, cerebral tumors can be regarded as a mix of abnormal cellular and vascular elements. And such histological and metabolic differences between tumor and normal brain tissues may cause changes in the CBF or cerebral blood volume, and that has been utilized over here. Now, they've mentioned the importance of their work over here. This is the first report utilizing bold resting state fMRI and ICA to delineate both central nervous system and non-CNS tumors preoperatively. So what they did with ICA is, first of all, the RS fMRI data is undergoing decomposition. That is, independent component analysis used to extract spatially independent components which could represent different brain networks or sources of noise. The main goal here is to identify components that correspond to tumor-related activity or alterations in brain networks caused by the tumor. Now, there is this entity called total number of components. Before I make this more complicated, let me simplify this a bit. So imagine you have a big messy ball of spaghetti where the different noodles are tangled up and each noodle is a different color representing different types of brain activity. Now let's say the red noodle represents activity from a brain tumor, while the blue, green, and yellow noodles represent other normal brain activities. So it's hard to see the red noodle clearly because it's mixed up with all the others. So in this study, scientists use a special untangling tool called ICA, like a special fork that helps separate out the different colored noodles so that each type can be seen more clearly. Now, there is a tricky part to it. We have to decide how many noodles to pick out to get the clearest view of the red noodle. That is the tumor activity. So if we pick too many or too few noodles, you might not see the red noodle well because it's tangled up with the other colors. The number of noodles we pick is the TNC, otherwise known as like total number of components in the study. The number of components used in this decomposition is a crucial parameter because it dictates how many independent components will be obtained. Different TNCs settings can produce different results, as varying the number of components may affect the ability of isolate tumor signals from brain activity, from other brain activity or noise. Now, the scientists, because of this, as in the TNC should be optimized for tumor component identification. So the study developed an algorithm to automatically determine the best TNC setting. So scientists came up with a smart way to figure out exactly how many noodles they pick in order to separate out the red noodle as clearly as possible, according to my example. So once they do that, they can learn a lot more about the red noodle, like how long or thick it is. Otherwise, which helps them to understand the tumor's effect on the brain. So this ICA is basically used to untangle the brain's activity and find the part that shows the tumor's impact by picking just the right number of noodles to separate, according to our example. Hope everyone's fine with the concept. By applying ICA with the optimized TNCs, the researchers aim to identify the independent component that most accurately corresponds to the tumor-related brain activity. I'm just re-emphasizing on it. And they've given an illustrated case over here. They adopted an automatic template matching algorithm named discriminality index-based component identification. And then they've plotted it over a graph in here using their steps. You can go through it if you want to, but the basic principle remains the same. Now, the thing is, the success rates are 100%, 100%, and 93.75% for glioma tissue detection for the three centers in which they did, actually. That is what's given in here. And 85.19% for the musculoskeletal tumor detection. They propose that the high success rate could come from the previously overlooked ability of bold RSFMRI in characterizing the abnormal vascularization, vasomotion, and perfusion caused by the tumors. Now, there were failed cases too. And different centers obviously have different mean optimal TNCs. Significant individual based variability was there. And tumor, like the algorithm, selected a component that partially covered the tumor region as confirmed by visual evaluation. This thing was also there. However, the visual inspection did not find any other component that was better than the automatically selected component. This means that the algorithm worked well, quite well actually. And there are a few limitations to the study. The sample size of tumor patients from each center needs to be increased further. Then it needs to be validated on more cases and best ICA decomposition based on the results. By matching it with the template was done. Some existing ICA algorithms are also promising and they can be used to. The DICA algorithm which they used only use spatial information for tumor detection, but it ignores the informative temporal frequency information. This is another drawback. So that's all from this article. Thank you everyone for your patient listening.